Okay guys, get ready for an intense revision session for the KCSE 2024 Biology Paper 1. So I'm not just going to go through the questions, I will break down each question, thoroughly discuss the concepts and ensure you understand the underlying principles. By the end of this session, you will feel confident enough to tackle such an exam by yourself with ease. Let us start. Question number 1. The following diagram shows an apparatus used in ecological studies. Part A. Name the apparatus. This is the pooter. Part B. Why is glass preferred in the making of the part labeled A? So glass is transparent. That means that you can easily look through it in order to observe the specimen. In order to study the features of the specimen and so on. And that is the reason why a lot of apparatus are made from glass. Question number 2A. Name the kingdom whose members are all microscopic. This, of course, is kingdom monera. This is the kingdom to which bacteria belong to. Part B. State 2 diseases that are caused by organisms belonging to the kingdom named in 2A. So essentially, we are looking for diseases that are caused by bacteria. And we have quite a few. So you can write cholera, typhoid, tetanus, meningitis, pneumonia. All of these are correct. By the way, fun fact, did you know that out of all the bacteria that are present, less than 1% of bacteria are actually harmful? I know, surprising, right? So that means that the vast majority of bacteria are either beneficial to us or completely harmless. So this is actually a case whereby bacteria have the worst PR possible because all it took is just a few bad strains of bacteria to give the whole kingdom a viral reputation. Question number 3. During a microscopy practical, the following materials were provided. Okay, so we have a temporary mount of an onion epidermis and a transparent ruler. Part A. State the aim of the experiment. So the aim, of course, is to estimate the size of a cell. Now let me explain this. I'll start off by explaining the materials that were provided, and then we're going to go back to the actual question itself. So number 1, we have an onion epidermis. So this is simply the outer layer of an onion bulb. Now this is used in a lot of microscopy practicals because it provides a clear, thin tissue that is ideal for microscopic observation, for observation under a microscope. Now, the reason for this is because the cells that are present in the epidermal layer of the onion tend to be large, they're visible and uniform, so you can easily observe the cellular structures that are present within. I'm talking about structures such as the cytoplasm, the nucleus, the cell wall. These can be easily visible when observed under the microscope. Now, the onion epidermis has been placed on a slide, and this is what is termed as the temporary mount. So it has been prepared for immediate observation, okay? For observation under the microscope at that particular time and not for long-term use. Now, moving on to the next material that was provided, this is the transparent ruler. So the ruler is used to measure the field of view. Now, in case you're wondering, what is the field of view? This is simply the area that is visible under the microscope as such. And this usually appears as, circular, okay? So if you want to estimate the size of a cell, how do you go about it? Number 1, you're going to take the ruler, okay, place it on the stage. Now by doing so, you are going to measure the diameter of the field of view. Now remember, the field of view appears as a circle. So you're going to measure the distance from one end to the other, and this is what is referred to as the diameter. Next, you're going to count the number of cells along the diameter of the field of view. So for example, over here, you're going to count the number of cells from this end to that end. How many cells do we have? There you go. Okay, so this is just an example. The number of cells can differ depending on the specimen that is used. Now the last step, this is whereby you will estimate the size of one cell by using the following formula. So the size of one cell is given by the diameter of the field of view. This is going to be in micrometers over the number of cells. And there you go. Moving on to question number 4. The following diagram represents a specialized animal cell. Part A. Identify the cell. Okay, let's look at the cell. Do we know what this is? Yes, this is the sperm cell. Part B, Roman 1. Name the cell organelle that is likely to be found in abundance in the part labeled F. So essentially, they are asking us in the part labeled F, which organelle would be plenty? Now, this organelle will be tied to the function of the sperm cell. What is the function of the sperm cell? It needs to fertilize the egg cell. But for it to do so, it needs to propel itself to where the egg cell is in the fallopian tube. So that means it requires a lot of energy. And therefore, it needs to have plenty of mitochondria. So the cell organelle that is going to be abundant is going to be the mitochondrion. Roman number 2, explain the answer in 4b1. So mitochondria produce energy that is required for the propulsion of the sperm cell. Question number 5. Name 2 components of blood that are present in the tissue fluid. Okay, so tissue fluid is simply the fluid that surrounds the cells in the body. Now, when it comes to the components of blood, are we going to have the blood cells present? Yes and no, depending on which blood cells we are talking about. Red blood cells, no. These are going to be absent in the tissue fluid. Okay, platelets too. Platelets are also not going to be present in the tissue fluid. Now, when it comes to white blood cells, are white blood cells going to be present in the tissue fluid? Yes. Some white blood cells will actually be present there. Now, the reason is because white blood cells defend our body against pathogens. These are microorganisms that cause diseases. So they need to be present where infection is. Now, one characteristic of white blood cells is that they are very flexible. So this allows them to squeeze through narrow spaces in the body in order to reach the sites of infection. So if the site of infection is in the tissue fluid, white blood cells will definitely be there. Okay, so white blood cells is not going to be included here. Okay, because yes, some of them might be present, some absent, so no. Now lastly, plasma proteins. Now when I talk about plasma proteins, I'm talking about the proteins that are normally found in the blood plasma. For example, fibrinogen, albumin, and so on. Now these are not going to be present in tissue fluid in large quantities. And this is because their molecules are too large to pass across the capillary walls and into the tissue fluid. So they mostly remain within the blood capillaries and do not move out. Question number 6. Name the structures in plants through which the processes of transpiration and guttation occur. Okay, so transpiration, this occurs mostly through stomata, but it can also occur through the cuticle and lenticels. Guttation occurs through hydatodes. Sawa? Okay. Now, what are hydatodes? So hydatodes are specialized structures. They are actually pores, okay, that are found in the leaves of some plants. So they are typically located on the edges of the leaves as such. So these pores allow the plants to lose excess water in form of droplets. Droplets, yes. So this is actually one of the main differences between transpiration and guttation. So when it comes to transpiration, this involves the loss of water, yes, but in form of vapor. When it comes to guttation, you're going to have water being lost in form of liquid droplets. Now, another difference between the two is the structures through which these processes occur. In transpiration, mostly through stomata, and in guttation, through hydrothods. Moving on to question number 7. Name 2 classes of the phylum Arthropoda that have a cephalothorax. Now, what is a cephalothorax? This is essentially a body region that is formed when the head and the thorax fuse together. So you're going to have such animals having 2 body parts. They will have the cephalothorax and the abdomen, of course. Now, this is found in class Crustacea and class Arachnida. Question number 8, part A. Name the source of hydrochloric acid in the human alimentary canal. This is the gastric glands. Part B. The following diagram shows a process along the mammalian digestive system. Roman number 1. Name the process. Okay, now let's look at the diagram. So if we look at the diagram, we can clearly see that we have this ball, it's called the bolus, and it's moving. So we can tell by the direction of the arrow that it's moving from one point to the other. Now, for this explanation, I'm going to start from the beginning of the digestive system. Sawa, tulisema this is revision, right? So the beginning is the mouth. Now, in the mouth, when the food is physically broken down through chewing, it's then mixed with saliva and then rolled to form a soft, moist ball. This is called the bolus. This is then pushed to the back of the mouth by the tongue and swallowed. So it moves through the esophagus and into the stomach through the process of peristalsis. So what is peristalsis? This is a process whereby you're going to have rhythmic contractions of the muscles that are present in the digestive tract. Pause. In our digestive tract, we have muscles that are located on its walls. And these muscles are capable of contracting and relaxing rhythmically. Now, they are going to be present in certain parts of the digestive tract, such as the esophagus, the stomach, the intestines, and also a few other parts. Now, these contractions that are as a result of the muscles are very important because they facilitate the movement of food along the digestive tract. And in short, they propel the food forward, moving it from the start to the end of the digestive system, which is the anus. Now, another role of peristalsis is that it also allows for the mixing of food. So, for example, in the small intestine, peristalsis ensures that the food is thoroughly mixed with the digestive juices, okay? And it also allows for the absorption of nutrients. So, win-win situation. On to our next question. Name 1 blood disorder caused by gene mutation. Okay, so we have sickle cell anemia and we also have hemophilia. Now, if you're an upcoming candidate, these are the disorders that you are going to learn in detail in the first chapter of biology in Form 4 called genetics. Such a fascinating topic it is. Okay, next question. Name the stage in meiosis where each of the following processes occur. Part A, formation of spindle fibers. This occurs in metaphase 1, metaphase 2. Okay, part B, disappearance of the nucleolus. This occurs in prophase 1 as well as prophase 2. So either of these two is correct. Question number 11. The following diagram represents part of the human male reproductive system. Part A, name the part labeled G. Okay, let's look at the information we have. Part G, that is definitely the prostate gland. Part B, state one function of the structure labeled H. What is H? That is the seminal vesicle. So the seminal vesicle secretes an alkaline fluid that provides nutrients to the sperm. Part C. How is the structure labeled J adapted to its function. Now, if you look at J, by the way, you can clearly tell that it's highly coiled. It looks to be coiled, right? So it's highly coiled to increase the surface area for the storage of sperms. Now, I want to say this. At this point, I'm not going to go into detail regarding the male reproductive system. The reason is because I already have a video discussing in detail the different structures present and the role they play. So if you're interested in this fascinating reproductive system, check it out. I will link it below. Moving on to question number 12. How do the following structural modifications in plants minimize the rate of water loss? Okay, so we are talking about reducing the rate of transpiration because this is the process through which plants mostly lose water. Part A, leaf folding. So by folding the leaf, what happens is that you reduce the surface area of the leaf that is exposed to environmental conditions. I'm talking about light temperature. So by reducing the exposure of the leaf to such conditions, you are going to reduce the rate of water loss through transpiration because most of the water that is lost through stomata, and stomata are present in leaves. Part B, sunken stomata. Okay. Now, this is something that I actually want to explain in detail because when it comes to sunken stomata, I find that some students tend to be confused on what sunken stomata are. So let me briefly explain. Now, stomata in general are located on the epidermal layer of the leaf as such, but when it comes to sunken stomata, these are special in that they are located deeper within the epidermis of the leaf. So by doing so, they end up creating, a depression or a pit, yani kashimo hivi, around the stomata. So I want you to remember, stomata are essentially just pores. They're just openings that are found on the surfaces of leaves. Now, in most cases, stomata are located on the upper and lower surfaces of the leaves, okay? But when it comes to sunken stomata, they are located deeper within the epidermis. So it ends up creating a pit above the opening. Now, these pits are very important because what happens is that you're going to have water vapor accumulating within these pits, okay? It's going to deposit within these pits, right? So at the end, this will end up reducing the concentration gradient. Let's pause there. What is the concentration gradient? This is simply the difference in the concentration of molecules between two regions. So for example, if you have two regions, okay, and in one region you have more molecules, another region you have fewer molecules, what will happen is that you are going to have a concentration gradient because the two regions have a difference in the concentration of molecules that are present. So if a concentration gradient is present, then that means movement of water molecules will occur. Now, the larger the difference, the better, because the movement of water molecules is going to be faster. Now, coming back to our pits, when these pits accumulate water vapor, you're going to have a scenario whereby the concentration gradient is lowered because you're going to find that there's a lot of water vapor accumulating within the pits, a lot of water vapor within the leaf itself. So the concentration gradient reduces. This makes it harder for water to be lost to the surrounding. So what happens next? It's going to reduce the amount of water vapor that is lost. Now, in case you're wondering, is this a good thing? Yes, it can be. If you have plants that grow in areas where, the temperatures are quite high, they cannot afford to lose a lot of water. I want you to remember that when it comes to transpiration, this is like sweating to us. When we sweat, we lose water. So in the case of plants, they also lose water through the process of transpiration, but there needs to be a balance. So if they lose too much water, they will end up wilting. So what happens is that in such plants, as a mechanism to reduce the amount of water that they lose, they have sunken stomata. By the way, if you find transpiration to be challenging, I have videos explaining this in detail. Check it out if you will. Now, moving on to question number 13. State 2 reasons for the absence of complex excretory organs in plant. Now, this question wants you to discuss the reasons why plants don't need to have complex excretory organs. Plants don't. They just have simple excretory organs, okay? So why? What is the reason why plants don't need to have complex excretory organs? So number one, plants have less metabolic activity and therefore they produce less wastes, okay? So yeah, they don't need to have, complex excretory organs because the amount of waste that they produce is less compared to that of, let's say, animals. Now, number two is that some of the waste materials that are produced by plants are recycled, they are reused. Now, a very good example is carbon dioxide. So, carbon dioxide is a waste product that is produced during respiration, but plants are able to recycle some of it and reuse it for photosynthesis. So, it's produced as a waste product during respiration, but plants use it for photosynthesis. So, win-win situation. Now, the key word here is some. Not all waste materials produced by plants are recycled, just some, such as carbon dioxide, water, and even oxygen. Remember, oxygen is a product that is produced during photosynthesis. So plants can then go and recycle it and use it, during respiration. Okay, now the next point is that the waste products that are produced by plants are less toxic. Less, not non-toxic, no. As long as you have waste products produced, they are going to be toxic, especially if they accumulate to a certain amount. But the waste products produced by plants are less so, and therefore they don't need to be eliminated quickly. Moving on to the next question. State the significance of each of the following characteristics in mammalian gaseous exchange structures and surfaces. Part A, the presence of rings of cartilage in the trachea. So the trachea is surrounded by cartilage rings. Why? To prevent it from collapsing. Part B, numerous blood capillaries lining the lungs. So these blood capillaries are numerous, there's so many, and the reason is to maintain a steep concentration gradient for the rapid diffusion of respiratory gases. Remember, when we talk about respiratory gases, we are simply referring to oxygen and carbon dioxide. Question number 15. Part A. What is the most appropriate method of estimating the population of black ants in a school playing field? Now for this, you can use capture-recapture method. Part B. Why are shorter food chains advantageous in an ecosystem? Now, in any ecosystem, what will happen is that you are going to have loss of energy as you move from one trophic level to another. Now, the energy is lost through processes such as respiration, excretion, and such. So when you have shorter food chains, you end up having fewer levels between the producers and the top consumers as such. Which is good because this means that less energy is going to be lost as it passes through the system. So it's going to make the whole process of energy transfer very efficient. Question number 16, part A. Using an example, define convergent evolution. So this is the modification of structures of organisms of different embryonic origin to perform similar functions. So the example that I'm actually going to use is the wings of bats and insects. Now, evolution is a topic that is discussed in Form 4. It's actually the second chapter in Form 4. Now, when it comes to convergent evolution, what happens is that you're going to have organisms that don't have a common ancestry. They're not closely related to one another. Let me give an example of insects and bats. So an insect or insects belong to phylum Arthropoda, right? Now when it comes to bats, bats are mammals. These belong to phylum Chordata. So these are two animals that belong to two different phyla, completely different. So they don't share a common embryonic origin, okay? They don't share a similar ancestor. But these two types of animals both have wings, and the wings allow them to fly. They perform a similar function for both the organisms. So you're going to have structures present in these two organisms that perform the same function, but the structures are different. The way they are composed or the way they are made up are different from one another, because at the end of the day, these two animals are not related to one another. Now, let me explain this in detail. So in the case of insects, you're going to have the wings extending from the exoskeleton. The tough outer covering that is present in insects. And these wings are going to be composed of cuticle. But in the case of bats, it's totally different. These are mammals and they will have the pentadactyl limb structure. So penta refers to 5 dactyl limbs. So this is a type of arrangement of bones whereby it ends up with 5 digits. Okay, 5 toes or 5 fingers, as in the case of humans, because we are mammals. Bats, being mammals, also have a similar arrangement, the pentadactyl limb structure. So do you see the differences in these two? They are very different. So they both have wings, okay, and they use them for the similar functions, but the way these wings are made up, their structures are completely different, because these two organisms, have different embryonic origins. Okay, question number 17, A Roman 1. Name the blood vessels that carries oxygenated blood from the heart to the rest of the body tissues. This, of course, is the aorta. Roman 2. State the role of the tricuspid valve in the mammalian heart. Now, we have two cuspid valves, right? We have the tricuspid valve and the bicuspid valve. The tricuspid valve is found on the right side of the heart. The bicuspid valve is found on the left side of the heart. By the way, if you find these two confusing, just use this BL. Bicuspid, left. So that means tricuspid is found on the right side. So the tricuspid valve is found between the right atrium and the right ventricle as such. Now what happens is that blood flows into the right atrium and then it flows into the right ventricle. Now once in the right ventricle, the walls of the right ventricle contract in order to push blood out of the heart. Now, when this happens, there needs to be a mechanism to ensure that the blood does not flow back. And this is where the tricuspid valve comes in. So when the blood has flown into the ventricle, the tricuspid valve closes up. Okay? So when the ventricles contract to push the blood out of the heart, it ensures that the blood cannot flow back into the atrium. So how do we state this? It prevents the backflow of blood into the right atrium when the ventricle muscles contract. Part B. Why are people with blood group O referred to as universal donors? Now, people who have blood group O lack antigens. They don't have antigen A, neither do they have antigen B. Therefore, in the case of blood transfusion, no agglutination can happen with the recipient's antibodies because there are no antigens to react with. Question number 18. The following diagram shows germination in two different seedlings labeled M and L. A Roman 1. Identify the type of germination shown in seedling L. This is hypogeel. And what is the reason for this? Because the cotyledon, as you can see, has remained below the soil surface. Okay, it's underground, so that makes it hypogeel. Part B. State one common function of the parts labeled K in seedlings L and M. So this is the cotyledon. It's used for the storage of food, okay? And this food is needed because it's going to be broken down, hydrolyzed in order to provide energy for germination to take place. Question number 19. The following diagram represents a longitudinal section through a phloem tissue. Part A. Account for the high concentration of mitochondria in the part labeled N. Okay. Now, the reason why you have a high number of mitochondria in the part labeled N, this is the companion cell, is so as to generate enough energy that is needed for the translocation of food substances from one sieve tube to another. So when you talk about translocation, this is simply the movement of food substances from one part of the plant to another. This, of course, happens in solution form. Part B. State one structural adaptation of the part labeled P to its function. Okay, so number one has cytoplasmic filaments which are continuous from one sieve tube to another. And this, of course, allows for the translocation of food. Number two, they have sieve pores, those for the passage of the cytoplasmic filaments. So the cytoplasmic filaments are able to pass from one sieve tube to another because of the pores, the sieve pores. And this allows for the continuous translocation of food from one part of the plant to another. On to the next question. The following word equation represents a metabolic reaction taking place in an animal tissue. Okay, so we have glucose. Glucose is being broken down to produce lactic acid and energy. Energy. You have energy being produced. So that means that this process is respiration. But which type of respiration is this? This is anaerobic respiration, whereby food substances are broken down in the absence of oxygen. So that means the condition under which the reaction occurs is the absence of oxygen. Part B. How does the size of an animal affect the rate of respiration? Now, when it comes to animals, the animals that are smaller in size tend to have higher rates of respiration. So if I had these two animals over here, I'm going to expect that to have a higher rate of respiration. Why? For the simple reason that it's smaller in size. Now, when animals are smaller, they tend to have a higher surface area to volume ratio. So this means that they lose more heat to their surrounding. Okay? So the smaller animal loses more heat to the surrounding. So that means that they need to replace the heat that they are continuously losing by producing more. They lose more, they produce more. Are you seeing that? So they end up having higher rates of respiration in order to compensate for the heat that they are losing. Now, when it comes to animals that are large, it's the opposite case. So they are going to have a lower surface area to volume ratio. So what will happen is that, yes, they are going to lose heat to the environment, but not as much, okay? So that means they don't need to produce as much heat as the smaller animals, because they don't need to compensate for much. Okay, moving on to the next question. Part A. How can sexual reproduction in organisms lead to the evolution of new species? Now, sexual reproduction occurs when you have two individuals. These individuals have gametes. The gametes are sex cells, okay? So the gametes come from two different individuals, and as expected, they are going to have different genes. Now, when these gametes fuse, they will lead to the formation of a new organism. Now, this organism is going to have, certain characteristics that are unique to them. And it's going to pass these characteristics that may be desirable to the next generations, okay? Until finally this leads to the establishment of a new species. Bear in mind, this is something that happens over a long period of time. Long. Okay. Alright. Part B, state the role of continental drift in the evolution of organisms. Okay, story time now. So according to this theory, a long time ago, and I mean a long time ago, there existed only one landmass. This was called Pangea. So the animals could freely move from one part to another. And then Pangea broke down into smaller parts that moved away from one another. They drifted from one another. So these smaller parts are what led to the formation of the present day continents that we know of. Africa, Asia, Antarctica, and so on. So when this happened, the organisms that had previously the same ancestry had to evolve differently over time. And this led to the development of different species. Question number 22. The following diagram represents an experimental setup used to investigate a certain biological process. Part A, Roman 1. Identify the biological process that can be investigated using the setup. Okay, let's look at our setup. We have a suspension that consists of glucose and yeast. Okay, so we're being told that the glucose was boiled and then cooled, and then yeast was added onto it, forming a suspension. Now above it is a layer of oil. And then we have this connected to a boiling tube containing lime water. So lime water is calcium hydroxide. This is used to test for the presence of oxygen. So we have yeast, we have glucose. So we are expecting that respiration will take place. Specifically, we are expecting anaerobic respiration to take place. Okay, this is also known as fermentation. Now, don't leave it hanging as simply respiration. This is wrong. As long as the setup makes it clear which type of respiration this is, then you need to specify. Roman number 2. Give a reason for the answer in 22A Roman 1. Okay, why are we stating that this is anaerobic respiration? What makes us so sure that there's no oxygen, over here? Because yeast can actually carry out aerobic respiration. So for them to carry out anaerobic respiration, that means that oxygen is absent. Okay, now the reason is because number 1, the glucose solution has been boiled. So by boiling, you drive out any air that is present within. Okay, so you're literally expelling any oxygen that is present. And then on top of that, you've added a layer of oil above the suspension. So you are preventing any re-entry of oxygen. So no oxygen means anaerobic respiration. Part B, write a word equation illustrating the reaction taking place in the experiment. So we have glucose being broken down to give ethanol, carbon 4 oxide, and energy. Part C, suggest a modification on the setup that would increase the rate of reaction in the conical flask. Okay, so how can we speed up the rate of respiration? Number 1, by increasing the temperature to optimum. Now this is respiration and it's a process that is controlled by enzymes. If you have optimum temperature, the enzyme reactions are going to be the highest. Okay. Now, number 2 is increasing the concentration of the yeast or glucose. Part D. Why is it necessary to cool glucose before adding yeast in the conical flask? Okay, so what happened is that the glucose solution was boiled. As we stated before, this is to remove any oxygen that is present. Then it was allowed to cool before the yeast was added onto it. So why did it need to be cooled? Why couldn't the yeast be added directly to the glucose solution when it was hot? Now, the reason is because yeast consists of yeast cells. Now, if they are exposed to high temperatures, what will happen is that you will end up killing the yeast cells. Now, by killing the yeast cells, you will also denature any enzymes that are present within the yeast. Okay? Remember, enzymes are sensitive to temperature changes. If you have temperatures above 40 degrees Celsius, what will happen is that the enzymes are going to be denatured. So that means if you have dead yeast cells, okay, and you have denatured the enzymes, respiration will not take place. So we will not have anything happening there. So in order to avoid that, we cool the glucose before adding the yeast. Question number 23. The following diagram represents a bone obtained from a mammalian axial skeleton. Part A. Identify the bone. This is the axis. Roman 2. Identify the part labeled R. This is the odontoid process. Okay. Now, part B. Name the bones that articulate at the points labeled S and T. Now, when we talk about articulation, we simply mean the point of connection between, two bones, okay? So, which bone articulates with the axis at the point labeled S? That is the atlas. At T, that is the third cervical vertebra. Now, I want to say this. If you're a pre-candidate or a candidate that has yet to cover this chapter, well then, let me set your mind at ease. This is a chapter that is taught in Form 4. It's actually the last chapter of the biology syllabus. So if you want to have a heads up, which is, by the way, is always good, then check out my videos under the playlist labeled Support and Movement. Okay, moving on to our last question. Is it our last, by the way? Yes, it's our last question. The following food web shows a feeding relationship found in a certain ecosystem. Now, if you want to take a look at the food web, kindly pause the video and do so. Part A. From the food web, identify the organism with the lowest biomass. This is the hawk. Roman number 2, the trophic level occupied by lizards. Okay, lizards occupy the third trophic level, and this is as a secondary consumer. Part B, name the type of feeding relationship between the lion and the cheetah. So the lion and the cheetah are predators. They compete with one another for prey. So the type of feeding relationship is competition. Specifically, it's interspecific competition. So interspecific competition is a type of competition that is found between two organisms that belong to different species, as is in this case. So the lion and cheetah are predators, yes, but they belong to different species. Roman number 2, the cheetah and the antelope. The cheetah is a predator, the antelope is a prey. So the type of feeding relationship is predation. Part C, explain the role bacteria will play in this ecosystem. Now, some bacteria are saprophytic. So saprophytic bacteria are involved in decomposition. They break down organic matter, and by doing so, they release nutrients into the ecosystem. And this, ladies and gentlemen, brings us to the end of this amazing revision session. Stay tuned for the biology paper 2 that will soon be uploaded. See you there.